Quite often when we go to hospitals, we end up with an infection. These infections can potentially be superbugs. A superbug is a bacterium that is highly resistant to antibiotics and are much tougher to kill than your standard strains of bacteria. Superbugs arise from many different reasons. One of the key reasons is the overuse of antibiotics and the misuse of antibiotics. Here in Australia, more than 170 Australians die from bacterial infections every week. Drug-resistant infections in hospitals are wreaking havoc and costing our economy more than $1 billion each year. The issue is compounded by a very real scientific problem. We're simply no longer able to find new antibiotics. Drug discovery is dominated by large pharmaceutical companies, but they have now left antibiotic R&D. So how can we address this issue? One major key is to uncover new molecular diversity. Molecules that have never been screened before, but could be the next new antibiotic. We currently have banks such as World Bank, seed banks, gene banks, blood banks, but there's no molecular bank. Until now, no one thought of working together as an international community to form a World Bank of molecules to address the superbug crisis. So COAD, the Community for Open Antimicrobial Drug Discovery, is an initiative where we can screen compounds from chemists worldwide. This will allow us to uncover new diversity that's just sitting on shelves waiting to be screened for antimicrobial activity. Pharma have always looked in one specific area for drug-like properties in their compounds when screening. But we're looking at space of compounds that have never actually been designed for antimicrobial activity. This will allow us to build a picture of what an antibiotic should look like. When collaborators are sending in compounds, we accept them in tubes or in plates. We then can reformat all the samples into a uniform tubing rack system. This allows us to use our high throughput robotic system to screen the compounds. In the primary screening, we screen against 7 key human pathogens, 5 bacteria and 2 fungi. If we get any hits here, we validate the potency of these hits with a dose response. We then also counter-screen against mammalian cell lines for cell toxicity. Through COAD, we'd hope to find the next new drug against superbugs.